Hello, this video will explain how to analyze time-lapse fluorescence data obtained via microscopy and convert the video data into a number. We shall see how we analyze the numbers in the next video. Now, once we have run the experiment, we save the data after using different microscopic platforms as CZI file or LSM file, sometimes the image sequences. CZI is proprietary Zeiss, so is LSM, by the way. The tool that we shall use to analyze this data is called Fiji. Fiji is just ImageJ, is a typical slogan of the software. The true reason for that is that the original program that is to be used is called ImageJ. It's been written, the first version was 20 years ago by Wayne Rasband in NIH in United States. Fiji is like a repack of ImageJ, ImageJ plus various plugins that allow you to analyze things better. Where do you get Fiji? First of all, I have my own distribution with all the plugins. Alternatively, you can just type Fiji ImageJ. Downloads will take you exactly to the downloads of the software. The software does normally not require installation, but it may require Java runtime environment, JRE. If you don't have the JRE and don't want to install it on your computer, select the no JRE version. That one is going to be a bit bigger than the rest, but it doesn't matter. You can just download, save it on your hard drive, and run from the hard drive by clicking the command file. The command file for that would be that ImageJ Win64exe. So it has been installed. It's just essentially copy pasted. Once you run Fiji, you get a window like that. There's one feature that is not readily enabled, and we shall need to get it running on our system. If we go to Help, then Update, it'll start alone. Updates, it helps to update from time to time. Sometimes the update doesn't improve it, but actually makes things worse. Nevertheless, we have arrived to this message, and we are tempted to click Apply Changes. But what I kindly ask you to do is to click Manage Update Sites instead to start with. These are the sites from which it updates its material, and you, in your copy of Fiji, will need to tick one of these boxes. Currently I have ImageJ, Fiji, Java 8. Yes, I'm talking about this box, BIG EPFL. Yes, could you please click that? Bioformats should also be clicked, but I think bioformats is there by default. Apply and close. I click Apply Changes. Please restart. I will close it and I'll restart it very happily. I have a shortcut for it, but in principle you can go on the location of that file. Just double click it or click it, depending on your system. I run Fiji again. and hopefully it loads. And I will make sure that I have the needed plugins for that. That will hopefully be installed using this maneuver. If they aren't, you can run update once again. There's a good chance that it did not pick up the update for that particular site that we have specified. It's to pick it up. I must say it takes a bit of time for it to load on this occasion. I'm again slightly worried, but not too much. There you go. That's Fidget. What you need to check before doing anything: click plugins, registration, and in registration it needs to have these two toolboxes, Stack Reg and Turbo Reg. These two toolboxes, as you may have guessed, come from BIG EPFL. That's the university, I think. That's the scientists working on there generated that, and these two are updated in that manner. Right, let me show you now we've started Fidget successfully, let me show you how to analyze simple calcium recordings. This is calcium recording, doesn't matter what it's about. It says MGComp6F, LCTR, right, AMP knockout, date, and the E1853. It doesn't matter in principle what it means, but just as a matter of fact. MGComp6F, M stands for mouse, GComp6F is calcium sensor, reports intracellular calcium. LCTR means that left control. R AMP knockout, right knockout of AMP. That is, we'll have two samples, one will be control sample, the other will be AMP knockout. That's the date, and that's the experiment number. I number my experiments so I can easily refer to them. I dragged the file onto my ImageJ window. ImageJ bioformats plugin suggests me the following options. I think I can have auto scale option in here. If I click OK, it will ask me how many series I want to import. What are these series? Well, console first of all suggests different errors of import. So far these are not errors but just warnings. The file contains more than one series. That is, the data was acquired in two different populations that were imaged in a pseudo-simultaneous manner. We have an automatic table that moves from one spot to another. In two spots on this table within a special perfusion chamber, there are two different types of objects, two different types of cells, and they were imaged continuously throughout a certain time. Solutions were perfused. The reason why for imaging 2, well, first of all, your throughput is higher, and you can compare objects that have different conditions in them. So we would like to ideally analyze all the objects, all the series. They are quite big, I must say. You can see that it works hard to load them. You can barely see anything in here. There are two files, 1 and 2. I click that and I click up. That's number 2. I click that and I click up. That's number 1. Let us do the following thing. You can see some green dots. At some point they spark. At some point they kind of oscillate. In order to give you a taste about the green dots, you can select this elliptical tool around that, turn around that, and then you go image stacks, plot z-axis profile. and it will plot the grayscale profile, the mean grayscale value of each of these frames within this area. If you click live and play around with this area, namely you move it around, the place a little bit, you will see that your display is changing, reflecting calcium dynamics in this particular spot of the image. I will try to persuade you that the spot corresponds to this particular cell. See, I can do that. I can go around. Right. Before we dip into analysis, we probably need to do two things. We definitely need to do two things. We may want to correct for motion a little bit. Firstly, and secondly, we would want to automatize the data acquisition. The reason is that obviously I could drag this region of interest, this little ellipse, all around and collect these curves from each point in the image, but it will take me a whole day doing that. So we need to do this more automatically. Firstly, let us correct for any motion artifacts. What are these motion artifacts? The recording is long and cells move throughout the recording a little bit. Therefore, we will prefer to ensure that this motion does not contribute to changes in our fluorescence intensity. How do we do that? Image stacks. Tools. Concatenate, I think. No, I'm lying to you. Image stacks, tools, combine. Image stacks, tools, combine. We have two stacks. Stack number 1, stack number 2, all straightforward. If we press OK, they'll make one stack, which will be twice as wide. There you go. Looks small, but actually quite wide. You can barely see anything in there. Can this be improved? Image, adjust brightness contrast. And you can, in principle, you can drag venue to that and make it brighter. I would not overdo on the scale because there's a chance that you will saturate your image in this way. You see, that is a sign of saturation already. What we do now to correct for the motion, we do two following things. They may not look very logical to you, but nevertheless. I have a macro that has macro is a set of very simple commands that I will be using, which is quite helpful. I'll take this image, which has green stuff, and I will, around the image, I will make a black border. A little black border, just in case one of the bright objects touches current borders that I have, because the correction does not work very well. When your objects touch the borders, I will ensure that all the objects are enclosed in the borders. Look, I've selected these commands: get dimensions and width and height, run canvas size. I select it, and then on my PC I right-click run command and run selected code. You will see that this will change the size of the picture. It will get slightly bigger. And the next step, I will try to find the moment when there was some kind of motion in here. I would adjust my position at this moment, and I'll do two things. I'll convert image from 16-bit to 32-bit. That was quick. Now, and next step is going to be very long. Plugins, registration, stack rag. Click. I select fine. Transformation. And I click OK. After that, I go and make myself a coffee, tea, buy myself some sweets, or and so on and so forth, because this will take quite a bit of time, depending on your PC. You can see we have 2150 frames altogether. It seems to be doing several frames per second, so it's not going to be like 1 hour, but it is quite slow. What it does, the process is called registration. What it does effectively is aligning every image with its predecessor, using the predecessor as a pattern. So in case there was some motion, the motion would be, in case, for instance, has moved laterally, it will try adjusting that, compensating for the motion in this way. I'll pause the video in the interest of time, and then I'll get back when the registration is over.